So maybe the most important part about understanding the amino acids is appreciating how they convert into, or they are used, I'm sorry, not convert, but how they are used in order to produce peptides. So peptides are small condensation products of amino acids. So if we look at the top of this slide right here, we've got basically amino acid 1 and amino acid 2. Now, those two different amino acids, what they're going to do is they're going to undergo a condensation reaction where water is produced and a peptide bond, which we will get into the details of a peptide bond down the way, but a peptide bond is formed between those two individual amino acids. Now, one thing that's kind of worth noting about this is, well, what are you losing? You're losing a hydrogen and you're losing a hydroxide from these two different groups. So what persists and what is kind of the final product? One of the things that I'd really like you to appreciate about this is your final product's peptide bond does not have a positive charge. Like I said, we're going to get into a peptide bond later on, but the N-termini of your amino acid number 2, which I'm going to underline that one right here, that N-termini of amino acid number 2 does not have an overall positive charge. We've got our lone pair of electrons there. Okay, then our carboxylic acid at the end of amino acid number 1. Well, that is going to lose that carbon-oxygen bond in favor of forming a carbon-nitrogen bond. Like I said, we'll get into more of this down the line, but peptides are fairly small. Peptides are small, and these are kind of the building blocks ultimately for producing an entire protein. Now, peptide ends are not the same. Okay, so the numbering and naming starts from the amino terminus. And I've been using this term N-termini over and over again because it's something it's almost like second nature. You just use it so darn often. Here is the N-termini of our peptide. Then we have 1, 2, 3, 4 peptide bonds that connect all of these 5 different amino acids. Now, this is a good representation of what a short peptide looks like, where we've got all of our peptide bonds, we have the N-termini, we have the C-termini, and then we have all of our different R groups of these amino acids basically coming off of the side. Now, we're going to be drawing peptides, and that's a very important part about this class because of the fact that when you draw a peptide, it's almost like if you can draw a peptide, you can draw a protein. Would you ever need to draw a protein? No, you would not, because a protein has several hundred amino acids. But if you can understand a short peptide and the chemistry and the interactions that exist between the side chains of those short peptides, then that's going to really help in understanding protein interactions within themselves, as well as how proteins interact with one another. Now, proteins are comprised of polypeptides, as well as, so the amino acids make up one part of a protein, but they might also have a cofactor. You might also have a coenzyme. You might also have prosthetic groups or other post-translational modifications. And this brings back that process of mRNA to protein. That process is known as translation. So these are modifications to a protein after translation has taken place. Now, cofactors are going to be small non-amino acids components, metal ions, or organic molecules. Coenzymes are organic cofactors. For instance, NAD plus in the enzyme lactate dehydrogenase is one. Prosthetic groups, maybe the most well-known prosthetic group is heme, which is at the center of hemoglobin and myoglobin. It's essential for binding to oxygen and subsequently the transport of oxygen. Now, we've been talking about proteinogenic amino acids, those amino acids that are integrated into proteins, but there are other amino acids that are uncommon. So those that are not incorporated by the ribosomes. One exception to this is selenocysteine, which is basically a cysteine residue, but rather than having a sulfur, it's got a selenium atom there. Now, uncommon amino acids in proteins arise from post-translational modifications. These are generally reversible modifications, especially phosphorylation. Phosphorylation is just the addition of a phosphate group. So add to a protein, specifically to something like a serine or a threonine or histidine, add a phosphate group to any of those. That can be added, it can subsequently be removed if needed to. And you can kind of think about the addition of a phosphate group as almost like a light switch. When you add it to a protein, well, that turns that protein on, it activates its activity. Whenever you remove it, maybe that inactivates it. But yeah, so there we go. Modified forms of amino acids, we've got a number of them here. So these are the results of modifications, and they're pretty dramatic for some of them. For like selenocysteine seems fairly simple. I'm going to highlight that one over here, because all that you're doing is you're taking that foundational cysteine residue and you're putting a selenium in there. If you compare that to something like 6N-methyl lysine, well, that's a methyl group that has been added to lysine. Compare that to pyrrolysine. Well, that's got a whole ring structure that's been added to the R group of lysine. So these all have a number of different functions, and yeah, they're kind of cool derivations of amino acids. Okay, so reversible modifications of amino acids, these are the ones that I want you to be more familiar with. The first three on this slide are basically the exact same. You just have to know what those amino acids look like, and, well, where could you put a phosphate group? You've got phosphoserine, phosphothreonine, phosphotyrosine. What do all of those residues have in common? They have a hydroxide group. You can make this a more high-energy molecule by throwing a phosphate group onto that serine, by throwing a phosphate group onto that threonine or that tyrosine. Others that you should be familiar with would be the omega N-methyl arginine, adenylyl tyrosine. So that's adding, so to a tyrosine residue, you're adding a phosphate group. So that's similar to a phosphotyrosine. But then to that, what you're doing is you're adding an entire nucleic acid. You're adding an adenine or an adenosine, which is the nitrogenous base, and the ribose sugar to that. So adenylyl tyrosine, you've added a phosphate, and then you've added this whole other thing on there. Glutamate gamma methyl ester and 6N-acetyl-lysine, these are all other derivatives of your common proteinogenic amino acids. Now, I want to take a moment to talk to you specifically about phosphoserine, phosphothreonine, phosphotyrosine. All of those residues are pretty unique in the sense that, well, you're taking something that had an R group that was arguably neutral. You had a hydroxide group. It was a little bit polar, certainly, and it could be involved in hydrogen bonding. But now what you've put in there in place is you've taken, think about it like this, you've taken an OH and you replace that OH with PO4. So you've taken an oxygen and a hydrogen and you replaced it with 4 oxygens and a phosphorus. In addition to that, now you've got 2 negative charges on there, because we're talking about this at a physiological pH. Well, if you talk about the interactions that a serine could be involved in, hydrogen bonding, definitely. It's a little bit polar, so it might be able to be involved in some other stuff. But then if you put a phosphate group there, well, that phosphate's going to change things dramatically. Now, all of a sudden, you've got something that couldn't be involved in ionic bonding. You've got something that has not just 1, but 2 full-on negative charges that can attract 2 positive charges. So the addition of a phosphate, one of the things that it does is it dramatically changes the interactions that that residue could be involved in. So it's a pretty stark difference. Now, this tells you basically a very dramatic table of polypeptide sizes and the number of them in a protein. So if we look at the top, we've got cytochrome C, which is 12,400 Daltons. The common unit for proteins is daltons or kilodaltons, so that's 12,400 daltons, or 12.4 kilodaltons. That is made up of 104 amino acids. Now, if we look all the way at the bottom, we've got Titan. It looks kind of like Tintin, but Titan. That is 2.9 million daltons. It is a protein that has 26,926 amino acids. Okay, so we've got this very dramatic ends of the spectrum. But then I'd like to draw your attention over to this, hemoglobin. Hemoglobin is a protein that's 574 amino acids and is made up of 4 polypeptide chains, compared to Titan, which is only 1 polypeptide chain. So we've got all these, like, hemoglobin is this protein's folded, it's going to wrap up with these two, and then these two are going to wrap together. So it's 4 things coming together to make one protein, one functional unit. Titan, well, Titan is several, let's see, about 40 times larger, 50 times larger, but is made up of only 1 polypeptide. And that's a pretty interesting thing. We're going to be talking about orders of protein organization later on, but just kind of keep this in mind as we move forward. Now, one thing that I'd like to come back to is each amino acid, although they vary, each amino acid has an approximate mass of 110 daltons. But like I said, they vary a little bit. 110 daltons, or 0.110 kilodaltons. Okay, so conjugated proteins are covalently bound to non-protein entities. For instance, lipoproteins, glycoproteins, which we'll talk about later on this semester. They're connected to lipids and carbohydrates, respectively. Carbohydrates serve as your mechanism for determining blood types. Metalloproteins are proteins that are almost permanently affixed to metal ions like iron, zinc, or calcium. And then we've got some examples of those. Common questions about peptides, common things that you need to think about if you're studying a protein. You need to think about what's the sequence and the composition. And by that I mean, is it made up of 1 polypeptide or is it 3 polypeptides? Then, what is the 3-dimensional structure? So what does that thing look like in 3D space? Does it look like this microphone right here? No, it doesn't. Chances are it's globular, it's spherical, but it probably looks like a bean bag. I haven't seen a bean bag in 20 some odd years, maybe longer, I don't know. But yeah, they have this kind of relatively spherical shape, but then it's kind of amorphous. How does it find its native fold? We're going to get into that in terms of, like, how does a protein go from a polypeptide to this functional entity? How does it achieve its biochemical role, and how is its function regulated? All of these things we're going to get into in a later date. But if you decide that you want to pursue protein chemistry or studying proteins, these are all the things that you kind of want to be concerned with. And these are all great questions. You know, it's neat to think about, well, what does it look like? And what is its 3-dimensional shape? But if you don't know where it's located within the cell, the rest of it doesn't really matter too much. Likewise, if that protein is supposed to be in the mitochondria, but ends up in the nucleus, well, it's going to run into some problems from a functional perspective. Is it going to be able to be involved in fatty acid breakdown? If it's in the nucleus and it's supposed to be in the mitochondria, no, it's not, because fatty acid breakdown takes place in the mitochondria, not in the nucleus.